In this video, we will discuss about role of biotechnology in agriculture. Now, how biotechnology is improving our today's agriculture. This will be discussed in detail. This is the second video. Link of first video will be shared in the description box. So, biotechnology plays an important role in agriculture. As we all are dependent on agriculture for our food, so this is also very important for the progress of the country, economy of the country. So agriculture fulfills all the requirements of human beings, and it also plays an important role in the production of transgenic crops. What are transgenic crops? We'll discuss in the next video. So we all living beings, plants, animals, human beings, we are dependent upon agriculture directly or indirectly. So plants is the source of food of human being as well as of the animal, whether they are vegetarian or they are non-vegetarian. Agriculture is source of food, feed, fibers, clothes, timber, and many other things. So, due to increasing population, biotechnology is playing an important role in the production of the agriculture. This is improving the crops, this is also improving the yield of the crops. So, what is biotechnology basically? This we have discussed in various videos. Link will be shared in the description box, but briefly I will again explain. Bio mean living, technology mean techniques. So, with the help of living beings and techniques, we are improving the crops, creating the crops called as genetically modified crops. These GM crops are also called as transgenic crops. Now, what are transgenic crops basically? Transgenic crops are the crops where we introduce a DNA from a plant which we want to introduce into the host. Supposing this is the foreign DNA which we want to introduce into the host. This can be cut with the help of specific enzyme, restriction enzyme. Again, a detail of restriction enzyme have been discussed in one video. Link will be shared in the description box. Now this cut DNA can be introduced into the host DNA. So this is the host DNA, blue color, and this is the red DNA which is foreign DNA or insert DNA. Now they can combine with each other, and if they form RNA and protein, means show their expression, then the plant is called as transgenic plant. So other ways, methods were selection, hybridization, protoplast fusion. These methods were used for the creation of the transgenic crops, but now this is the latest technology which is also called as recombinant DNA technology or genetic engineering, because we are introducing a gene into some other plant. We are introducing the DNA into the other plant. So this plant with the foreign gene or foreign DNA is called as transgenic plant or transgenic crops. So there are various methods for introduction of the foreign material into the plant cell genome. We can directly or we can do it with the help of vectors. So there are different techniques by which we can introduce foreign genetic material into the plant cell genome. First, it requires selection and isolation of the desirable fragment of DNA through a vector. Vectors like plasmids, cosmids, again a link is shared in the description box. We have discussed in detail in one video. So vectors, cosmids, YARCs, bags, they are actually vectors which are used for multiplication of the foreign DNA. Another technique is we can directly introduce the DNA into the plant cell genome. This can be co-cultivation, electroporation, and micro-injection technique. We can directly inject the DNA into the host tissue. This is called as micro-injection technique. Electroporation is with the help of electric currents. So we can create the transgenic crops or genetically modified crops. Now what are its application of the transgenic crops in the agriculture? One, transgenic crops or genes, they can help in improving the photosynthesis. We know that photosynthesis is a process which help in synthesis of the carbohydrate or sugars in presence of light. So this is called as photosynthesis. An enzyme called as rubisco, this plays a very important role in the photosynthesis. Rubisco is full form is ribulose 1,5-biphosphate carboxylase oxygenase. If its carbon dioxide concentration is more, so this part of the enzyme will work. If oxygen concentration is more, then ribulose 1,5-biphosphate oxygenase will work. Now, ribulose 1,5-biphosphate oxygenase lead to photorespiration. Photorespiration is respiration in presence of light that leads to loss of the yield. But photosynthesis lead to the formation of the biomass or help in increasing the yield. So we are interested in this part of the enzyme which will improve the photosynthesis. So we can modify the enzyme where only this part of the enzyme will work, right? And we can introduce into the plant where yield will be increased, where loss is due to photorespiration. There we can improve the yield by inactivating this part, which is oxygenase part. So genes have been transferred from pea plant to tobacco to increase the photosynthetic process and to increase the yield of the crops. Now this is the tobacco plant. With the help of ribisco enzyme, its yield have been increased, and this gene have been introduced from the pea plant. Another application is a role of biotechnology in making the plant resistant to biotic stresses. Now, biotic stresses are like if we are growing some wheat plant, right? Or we are growing maize crop. Supposing this is the wheat plant and this is also wheat plant. In between, other plants are also growing, right? Now, these plants will compete with this plant for yield, water, minerals. We don't want these plants, right? So either we can employ a labor to uproot this plant, or we can use herbicides which can kill this plant. Herbicides are the plants which will kill these herbs or herbaceous plant without affecting our crop, right? So we are creating our crops which may be resistant to the herbicides. So this can be done. Either the target molecule become insensitive or target protein should be overproduced. Supposing this is the wheat plant, right? In wheat plant, if we are spraying the herbicide, right, this will become resistant to this herbicide. Either it will produce the target molecule which is insensitive, or it is this protein inside the plant which is affected by herbicide is overproduced. So a pathway will be introduced which will detoxify this herbicide, which will not be affected by this herbicide. So we can create herbicide tolerant transgenic plants like soybean, wheat, rice, cotton and maize plant which can tolerate these herbicides, BT cotton, golden wheat and maize plant. Then insect resistant in transgenic plants. I mean, we are creating plants which are naturally resistant to the pests or insect. One of the best example is BT cotton. BT cotton, we have taken the genes from the Bacillus thuringiensis. This is a bacterium, right? So this gene has been introduced into the cotton, and cotton plant will become naturally resistant to American bollworm. So crop which is having this gene is called as BT cotton. So they will resist the bollworm infections, or they will resist the losses which are occurring due to the American bollworm losses. So we can say Bacillus thuringiensis, as we have discussed just now. Now this gene from this bacteria can be introduced into the plant with the help of a vector called as agrobacterium, right? Again, agrobacterium mediated gene transfer. Video will be shared in the description box. You can watch that video. Detailed have been already discussed how agrobacterium help in transferring of the genes and what is BT cotton. And this have been also discussed in the previous video of the salient achievements in crop biotechnology. So we can transform tobacco, tomato, cotton. Then there is a plant which were resistant to Manducta sexta, which is a pest of tobacco. I mean, this destroys the tobacco plant. So we are creating the plant which are naturally resistant to the pests or the we can say insects. So BT tomato, BT papaya, BT cotton. We are creating these varieties which are naturally resistant to the pests. Now next is how we can create resistant against abiotic stresses. Biotic is living, abiotic is the environmental stresses like drought, cold. If soil has more saline conditions, salinity, then metal toxicity, mean it may have heavy metals in the soil. So we are creating the plant which may be resistant to these. You can say abiotic stresses or environmental stresses, or we can say like this is drought, this is smoke. So these are different kind of abiotic stresses. First we'll discuss about resistance against drought, salt and heat, etc. Now first we will identify the genes which will make the plant resistant to drought, salt and heat. Then we will isolate those genes and we will introduce into the host plant. So number of genes which are responsible for providing resistance against these stresses have been identified, like heat, cold, salt, and heavy metal. And the protein encoded by these genes can be broadly classified into two groups. One, those protecting cells from dehydration. Dehydration is loss of water. So we can use osmoprotectants. Osmoprotectants are basically which helps in maintaining osmotic concentration in the plant or in the tissue. Proline, it is an amino acid. Glycine and betaine, and they help in maintaining the osmotic concentration inside the cell. Second are we can use the genes which are again stress responsive genes, right, and which can also be controlled with the help of transcription factors. So stress responsive genes will help in tolerating the plant to the stress conditions. Supposing we have this DNA, when this form RNA, this is called as transcription. For this process we need to have transcription factors, and we are creating the RNA for the stress responsive genes, mean which will make the plant resistant to the stress conditions. We can see again a different resistance against drought, salt, heat, etc. Plant can tolerate these stresses. So it has been shown that a cis-acting promoter elements are elements which control the regulation of the gene, expression of the genes, right? So promoter element called dehydration response element, DRE, plays an important role in regulating gene expression in response to these stresses. Now, what is gene expression? Basically, supposing this is DNA, this form RNA, then this form protein, right? So this is called as gene expression. Now, this is controlled by promoter element, regulator element, right? So this is called a regulation of gene expression. So this transcription process is also being controlled by transcription factors named as DERBIA, DERE1A, DRE2B. Now they will bind to DRE and activate transcription of these genes containing the DRE sequence in Arabidopsis. Right. Now, complementary DNA for these proteins have been isolated and used for the production of transgenic crops that were tolerant to these stresses like a drought, salt, and freezing. I mean, we can introduce the gene into the host plant which can provide resistance to these stresses, abiotic stresses. So we are successful in that also. Now, next application is role in chilling resistance. Like if they are freezing condition or they are cold condition, plants they are destroyed or killed, like rice, maize, soybean. They are destroyed by this chilling treatment. We can say if there is more freezing condition, they are killed. So we can change that condition. We can make the plant resistant to the chilling condition. Like we can create double bonds in the plants. For that we need to have enzymes called as desaturases. Desaturation is I'll just explain just now. Like this is the fatty acid which does not have any double bond. Fatty acid is part of the lipid molecule, doesn't have any double bond. So when these two hydrogens are removed, one double bond will be created. When these two hydrogens are removed, and another double bond is created, right? So these double bonds can be created. This is called as unsaturated fatty acid, and this removal of the hydrogen can be done with the help of desaturases. So desaturation of these saturated fatty acid, this can be achieved by at least 3 desaturases, and the gene encoding these enzymes have been used for producing transgenic plants with an increase in the chilling resistance. I mean, we can introduce these genes into the plant which will help in creation of unsaturated fatty acid. Like fatty acids can be palmitic acid, it can be stearic acid, right? Palmitic acid is having 16 carbon, stearic acid is having 18 carbon, which have saturated fatty acid, and we can introduce double bond in them, making them unsaturated fatty acid, which will prevent the injury due to low temperature. So we can introduce these genes, and which will help in creation of the enzyme inside the plant, desaturases, which will create the unsaturated bonds and which will make the plant resistant to the diseases. So this is all about this video. Now in the next video we will discuss improved yield of the crops, transgenic crops, how we can improve the nutritional quality of the crops like golden wheat, then golden rice, then transport and shelf life of tomatoes and other fruit crops can be improved. Then role in the floriculture, that is in the flower studies, right? So thank you for watching my video. If you like my video, please like, share and subscribe.